So this is day one of a 2-day learning session. Today's sessions that you'll hear over these next couple of hours are focused on unlocking EHE, a global momentum in EHE science. And David's going to take us through the agenda. He's on the board of directors of the EHE Foundation, lending his professional talents, which are many, to us, as well as his great skill in moderating and fielding questions from the audience and with our presenters. So David, thanks for being here, and I'm going to hand it over to you.
Thanks much. Thanks for having me again. And thank you, Dr. Rubin, for the great introduction that I think sets the stage for what we're going to hear about the rest of the day today and through tomorrow. And so we're going to have a few sessions today. We're going to start with session one, which is on the topic of advancing treatments at the bench and the bedside. And we're going to have three speakers who are going to speak back to back. We're going to introduce Dr. Tanas from the Department of Pathology at the University of Iowa, who's going to be speaking to us about human-derived EHE models, directly responding to some of the stuff we've been talking about with Dr. Rubin. We're going to then hear from Dr. Pasquali, who's going to talk about, and Dr. Pasquali is a clinical scientist and surgical oncologist at the National Cancer Institute Foundation of Milan. He studies new drugs to target these types of cancers. And lastly, in this first session, we're going to hear from Dr. Haddix with the EHE Foundation, who's going to talk to us about the EHE Biobank. After everyone's done talking, we're going to have a group Q&A session where we'll get all of the speakers back together to answer questions from the audience and address any other questions that arise. And with that, let me turn it over to Dr. Tanas.
So we've been working on developing some EHE primary cell cultures for tumors from the EHE Biobank. I'm going to run through how these tumors were derived. So these are 4 tumors that were obtained from the EHE Biobank. As you can see that 3 of the 4 tumors are primary EHE tumors, 1 was metastatic. I've listed the method of confirmation of EHE in addition to histologic confirmation, which Brian mentioned. The diagnosis was also confirmed by immunohistochemistry for CAMTA1 or anchored multiplex PCR. These are representative histologies for 2 of the tumors, EHE036 and 053. And you can see here there's a proliferation of these epithelioid cells arranged in cord-like architecture and embedded in this myxohyalin matrix. And both tumors diffusely and strongly express CAMTA1 by immunohistochemistry. And so from these tumors, these were received as cryopreserved samples. The samples were essentially minced into smaller pieces, treated with collagenase disphase, and subsequently grown in endothelial growth media. This is what the cell cultures look like under bright field microscopy. The cells are more epithelioid in some areas, but also spindle shaped at the periphery of these cell cultures. And you can see over here, this arrow over here is pointing at a residual tumor chunk in the cell culture, and these cells are basically emanating from that tumor. And so we validated expression of the fusions at an RNA level for 2 of the samples, and by PCR-based, RT-PCR-based methods. And as you can see here, 2 of these fusions in EHE1 and 36 demonstrate a breakpoint at the end of WWTR1, at the end of exon 2, and a breakpoint within exon 9 of CAMTA1. And as has been described before, these breakpoints result in CAMTA1 being expressed in frame. And so there's a functional peptide being expressed by these fusions. We also validated expression of the fusion in first 2 cases by quantitative RT-PCR using a Tachman-based assay and a probe that was specific for the breakpoint of the fusion in these 2 samples, and then also demonstrated expression of the fusion protein in these samples by Western blot. in an effort to characterize in vitro hallmarks of cancer in these EHE extended primary cell cultures, we looked at proliferation and anchorage-independent growth. We looked at proliferation via an MTT-style assay and used these primary endothelial cells, human pulmonary artery endothelial cells as control. And as you can see here, these 4 cell lines, these EHE extended primary cell cultures proliferate to a greater degree than the endothelial the normal endothelial primary cells. If we force these cells to grow in a suspended state on low attachment conditions using polyhema-coated plates. Polyhema is a polymer that forces cells to grow in suspension. You can see that cells derived from these tumors, these extended primary cell cultures, will form these spheroids in a suspended state, while the negative control, these human pulmonary artery endothelial cells do not. And so, again, showing that the cells are transformed. We can also do the same experiment in soft agar and see essentially the same thing, that these extended primary cell cultures will grow in an anchorage-independent state, but the negative control will not. We just did some really basic studies to see how these cultures responded to TEAD inhibitors. And as you can see here, with these 4 extended primary cell cultures, that they do respond to BT107, which is a pan-TEAD inhibitor from Therapeutics. And although the response to anchorage-independent growth was greater than that seen with proliferation, this is consistent with what we've seen with human-engineered cell lines, and that expression diffusions predominantly drive anchorage-independent growth, although proliferation is also one of the phenotypes seen. We then compared it with another compound, IK930 from IK Oncology, which is more of a TEAD1 focused TEAD inhibitor. And as you can see here, there's some differences in terms of how the cells responded to this TEAD inhibitor, and that proliferation does not appear to be affected as much as with BT107, although there is some abrogation of anchorage-independent growth, similar to what we saw with BT107. We then characterized at a genomic level these EHE extended primary cell cultures by whole genome sequencing, and we did see SNPs in these genes that were similar to what's been reported in these previous studies shown here on the right, just validating another way that these cell cultures represent what we know about EHE tumors. And if we do pathway analysis on SNPs, we'll see that ontologies, including changes in extracellular matrix, in particular, were enriched when we looked at these SNPs. We then used an approach integrating bulk RNA-seq and single-cell RNA-seq performed on one of the extended primary cell cultures. This is EHE01 that we performed the single-cell RNA-seq. One of the features that we noticed by single-cell RNA-seq is that as the number of mean reads per cell increased, the median genes per cell continued to increase as well. And usually at around 20,000, you'll start to see some plateauing here. And what was really interesting is that we continue to see discovery of unique genes identified per cell, which is consistent with the hypertranscription phenotype that's been described by Valerie Kusikov. So it was interesting to see some of those same features in an extended primary cell culture. We were interested to see how much drift there was from the cultured cells from the donated tumors, and so by bulk RNA-seq, we sequenced both the tumors, the cell cultures, and the tumors that they were derived from. We did not have any residual tumor from 053 for comparison, but we did it for the other 3. And as you can see here, there are some differences between the cultured cells and the donor tumors. Interestingly, the donor tumors tended to cluster together, as did the extended primary cell cultures, and these were, of course, different than the normal primary endothelial cells. if we look at compare the combined EHE, the differentially expressed genes from the EHE tumors and cell cultures, though, you'll see that approximately 50% of the differentially expressed genes overlap with those of the EHE tumors, and this was statistically significant. At the same time, although there was some drift from the extended primary cell cultures as compared to the EHE tumors, importantly, by pathway analysis, the main pathways were conserved. So especially with regards to PI3K and K2 signaling, ECM receptor interactions, Hippo signaling, they're all listed here, as well as in a more quantitative way in this table down here to the right. So the key features are maintained. We then asked the question, by single cell RNA-seq, are there different compartments in the EHE extended primary cell cultures? And this is unbiased clustering, comparing an early passage culture from EHE01 and a later passage one, in a later passage P17. And as you can see here, these are the 4 clusters that were identified. This first cluster over here was enriched for ontologies involving cell cycle progression, mitoses, while these 2 clusters, clusters 3 and 4, showed more of a senescent phenotype by pathway analysis. The second cluster over here showed features of endothelial vascular development as well as cell migration. And one of the things that we noted as we extended, as we continued to passage these EHE extended primary cell cultures, is that the growth rates diminished over time, consistent with the Hayflick limit. And this is something that's been observed from multiple investigators as we try to grow these human EHE extended primary cell cultures. If we go back and look at those clusters in the previous slide and look for genes that were involved in proliferation, stemness, and senescence, you'll see that in cluster 1, there is an increase in expression of PCNA, which is a marker of proliferation, as well as CD44. There was also increased expression of LIF that I'm not showing here that was also enriched in cluster 1. So this suggests that there's perhaps a proliferative stem cell-like compartment in this extended primary cell culture, whereas and you'll see that expression of these genes go down with the later passages. You compare passage 6 and passage 17. And then if you look at CDKN2A and P53 in the later passages, these genes are expressed at higher levels in the later passages, but particularly in clusters 3 and 4, which are these senescent cells off here to the periphery. And there's also an enrichment for these for mitochondrial genes, which are also associated with senescence. And so just to illustrate perhaps some of the difficulties that various investigators have noted with developing these EHE extended primary cell cultures, it does seem, at least preliminarily in our study, that there does seem to be a stem cell-like compartment in these EHE extended primary cell cultures with a proliferative and active proliferative component that demonstrates endothelial differentiation, but with time they will senesce, and perhaps in future studies we'll be able to delete CDKN2A or other tumor suppressors in an effort to prolong proliferation of these cells. We're also working with other investigators to develop PDX models. With that, I'd like to acknowledge the main person who worked on this project in the lab, Nick Scalora, who is a graduate student that graduated last summer. He's currently an assistant professor at Augustana College, which is a liberal arts college nearby in the Quad Cities, as well as Jillian, who's finishing up some of the validation experiments with these extended primary cell cultures. Would like to thank folks from the EHE Foundation, in particular Denise and Dr. Cogswell, as well as other collaborators and their sources of funding. Thanks for your time.
Thank you much. And with that, we'll have you step back and introduce Dr. Pasquali, and we'll see Dr. Tanas again after the next talks in a group Q&A session. And handing it over to you, Dr. Pasquali.
Yeah, hello everybody. Thank you for the nice introduction. I share my screen. Can you please confirm that you can see my slide in presentation mode?
The slides are showing and the audio is clear.
Okay, is it fine then?
It's excellent.
Okay, so thank you for having me in this presentation as representing the work of my lab, which has been starting by Dr. Zaffaroni before and continued by me and the team. So very glad to be in this meeting today. As a lab, we focus on two main issues, the paucity of EHE tumor models, particularly as Dr. Rubin mentioned before, the lack of patient-derived tumor models. So for us, that was an issue we want to tackle. And the other issue was the finding new treatment option for patients with aggressive EHE. So I will call that in this presentation high-risk EHE. So these were the two main unmet needs that we wanted to work at in the lab. We were lucky enough or good enough, I don't know, to generate the first patient-derived tumor models of EHE. And this was actually generated through subcutaneous implanting in mice, a sample from a primary EHE of a patient with an extremely aggressive disease. The tumor went through different passages in mice, and then we were able to generate also cell lines starting from the PDX. And here you can see that the morphology of the tumor of the PDX is similar to the tumor of origin. There was the TAZ-CAMTA translocation in the PDX and also in the human models. And then as we do routinely for our models, we show that the transcriptomic profile of our model resembles the one of the original tumor, and also a genomic analysis, so the DNA profile of the PDX was similar to that of the tumor. And finally, also the EHE cell lines is characterized by the presence of the TAZ-CAMTA fusion. So we were really interested in the high aggressive disease. So we started discussing with our medical oncologist, particularly with Dr. Stacchiotti and her team, about factors that might influence the severe inflammation and the symptoms that are there in the high aggressive disease. And exploring different cytokines, we came up with this GDF15. And we realized that it was released in cell culture medium from our EHE cell lines. And this was EHE, pretty much EHE specific. We didn't find that in other cell lines from other tumors. And we found GDF15 also in blood of mice with EHE PDX implanted. And this was not the same in healthy mice or in mice with other tumors. And these are just examples of other tumors. GDF15 is becoming very popular these days because recently it has been pointed out that it has an immunosuppressive role at the tissue level, and it might impair, the presence of GDF15 might impair the effect of immunotherapy. So there have been some trials blocking GDF15, enhancing the effect of immunotherapeutic drugs. And on the other end, GDF15 has been involved, circulating GDF15, and being involved in cancer-related cachexia. So one of the symptoms that is there also in the aggressive form of EHE. And there was very recently a trial with a drug targeting the GDF15 for the sake of resolving patient cachexia, and the trial was successful. GDF15 is involved in many different very relevant pathways in human cancers. Here, EHE is not reported, but I can tell you that each of these pathway is relevant for EHE. And also, for instance, Dr. Munir has done some work around PI3 PI3 kinase in EHE. So we convinced ourselves that GDF15 was relevant in the disease, and we went on, putting together this information of this cytokine with that of high-risk patients in our center as well as in other centers are treated with the mTOR inhibitor sirolimus. So we tried to make a connection between GDF15 and sirolimus, or at least we hypothesized that there was a connection between the two. So we started exploiting our models to test the effect of sirolimus on them, and this is our result on our cell lines. The red line is doxorubicin, the blue line is sirolimus, and here this graph showed that sirolimus is way more effective than doxorubicin in EHE cells, and the same applied to PDX. The black line is the control, so the tumor grow without treatment. The tumor similarly grow when it's treated with doxorubicin, but actually we show a delay in tumor growth when the treatment was sirolimus, and the higher the dose of sirolimus, the higher was the ability of the delay in tumor growth, and this is this line in dark blue here. And the right part of the slide simply represents that we saw effective inhibition of the mTOR pathway, including the downstream, when the mTOR inhibitor sirolimus was given to cell lines and PDXs. And this is the effect we observe at the morphological level. So normally in EHE, it's possible to observe necrosis, particularly in our PDX, but when we treat that with sirolimus, the PDX show also features of sclerohalinosis beyond necrosis, and these features were even more represented when the doses of sirolimus was higher, and this was in keeping with some level of response to sirolimus in our very aggressive human-derived, patient-derived xenograph of EHE. So we went on with this idea of looking at what happened to GDF15 when sirolimus is given to cell line and mice, and we observed that sirolimus was able to lower, you see the concentration of GDF15 in blue here compared to control, was able to lower the concentration of GDF15 in cell culture medium and in the blood of mice. And this was completely different with doxorubicin, which was not able to achieve that. So we tried to make sense of this through the downstream signals of the mTOR pathway, and we realized that was possible. We hypothesized that it was possible that transcription factors such as ATF2, 4, and 5 that are related to the mTOR pathway may be down-modulated by sirolimus. And in order to show this association, we realized that the transcription of GDF15 was lower when sirolimus was given to cells, and that was true both in cell lines and PDXs, also at the protein level, and this involved also the transcription factor ATF4 and 5, which are downstream signal of mTOR. So we went on inhibiting ATF4 and 5 with lab tools, which are called CIRNA, that can block the expression of ATF4 and 5, and we showed that actually the level of GDF15 went down when these siRNA were implemented. So these results support our hypothesis of sirolimus modulating GDF15 through the transcription factor ATF4 and 5. And we exploit a clinical trial, observation clinical trial, which is ongoing at our institution and at ICR London. And we saw that the GDF15 level was much higher in higher risk patients compared to lower risk patients, and definitely compared to healthy donors. And we confirmed this in an independent cohort of patients, and also to reinforce the data, we put the cohorts together. So we were quite persuaded that GDF15 correlate with presence of EHE and severity of EHE. These are just examples of some patients. We published this before. Here we saw that there was an increase in GDF15 level with the progression of the disease, and then the level of GDF15 GDF15 went down when treatment with sirolimus was started, and shoot up again possibly when the disease was progressing. So we were able to identify disease progression slightly before the performance of a CT scan, and we replicate the same scenario in some new patient actually, and this is just another example of GDF15 going up with the progression of the disease and going down with the use of sirolimus, which stabilized the disease. Luckily, very recently, a clinical grade test for GDF15 has become available. This is produced by Roche and is called ELIXIS. We did all our tests with an ELISA test in the lab, actually, so not in a clinical lab, but in a research lab. And then we redid all our tests using this clinical grade ELIXIS test. And here, basically, we showed that there was an high correlation between the level of GDF15 we found in patient and those that this new test point out, considering that this new test is much more reliable on low value or very high value. So this could increase the translatability of our work. Finally, something we work at is to realize whether the GDF15 could influence EHE cell phenotype, which means what happens if we block GDF15? Do we observe regression of the tumor or not? Or does the tumor change the tumorigenic properties such as cell growth, migration, invasion? Actually, the tumor did not. So when we inhibit GDF15, we didn't observe at the cellular level changes in cell phenotype. But when we went through the transcriptomic analysis to compare tumors that were knocked down or not for GDF15, we observed significant changes at the metabolic level involving different metabolic pathways, including also mitochondrial pathways. And this is something we are still developing and digging out at the moment. So I think this takes me to my conclusion. I think that for us, our research has some impact for the research on EHE, because we were able to establish the first, as Dr. Ruiz said before, the first human model of EHE, which we are now sharing with several scientists around Europe and North America, thanks also to the connection of the IAPTAS and TIT meeting where I've been invited last year and supported, thanks also to the foundation. We think also that our work has a clinical impact because it offers the opportunity to support an application, a formal application to EMA, the European Medical Agency, for the repurposing of the mTOR inhibitor sirolimus, which Dr. Stracchiotti and their team are carrying on. And this support also GDF15 as a possible companion diagnostic in this process. We are still working on the prospective study on EHE patients. I think tomorrow something will be presented by Dr. Frezza, and this is including GDF15 assessment and analysis beyond some other omic analysis and search for other biomarkers associated with disease and response to sirolimus. In a translational research perspective, our ongoing work now is finalized to associate GDF15 with patient outcomes, response to sirolimus, other profiling of primary tumors, which we are currently doing. And on the preclinical research side, since we have been generating the models and after doing the analysis that I show you, we started a bunch of different type of work. We're working, as I mentioned, on GDF15 and EHE metabolism, particularly in the interaction with the mTOR inhibitors, and also considering the effect of the TAZ-CAMTA fusion for the metabolism. We are working at other circulating markers other than GDF15, such as microRNAs, hormones, other cytokines to predict EHE aggressiveness and response to mTOR inhibitors. We are also doing other drug screenings, evaluating other drugs. Some are traditional chemotherapeutics that might be used straight away in patients with a repurposing aim, and other targeted therapies such as the TEAD inhibitors in our models. And also we are committed in the generation of new models and in other ways to model, to use actually other methodologies to model our EHE PDX and cell lines. I'm very thankful to our lab, to Valentina Zucco, who generated the models. Alessia was doing a PhD on that. Dr. Zaffaroni started the work on EHE at our lab some time ago. Silvia Stacchiotti in the medical oncology, who is interacted a lot with us, as well as all other collaborators here in Milano, and people at Royal Marsden and ICR who generated this work with us, and of course to the EHE Foundation that is supporting our work. And thank you for your attention.
Thank you very much, and we'll see you after the next presentation for the Q&A session. Tremendous amount of progress from your group, very exciting. And with that, I'll introduce Dr. Haddix to speak about the EHE Biobank.
Okay, am I live?
Your slides are live and your audio is great.
Fantastic. Okay. Hi, everyone. My name is Seth Haddix. I'm a recent addition to the EHE Foundation team. I'm also a postdoctoral bench scientist. That's my day job. And as a researcher, I have focused on the delivery of cancer therapeutics and studying rare disease. At the EHE Foundation, I've been working very closely with Patty Cogswell, who has developed and managed the EHE Biobank since 2020, and I'll be speaking today about the function and progress of the biobank. I've been working with Patty for about 2.5 years, managing biobanks for multiple foundations, and I've benefited tremendously from her guidance. And I'm excited to talk to you about what the EHE Biobank is and what it can be used for. There we go. So the EHE Biobank started consenting patients for inclusion in 2020. The biobank has undergone extensive scrutiny by an independent review board to guarantee that all its activities are ethically and highly rigorous. The biobank acts as a physical centralized repository where donated samples are held, and it also stores some basic clinical information from patients who donate, such as date of diagnosis, disease stage, that kind of thing, which can be linked directly to the specimens because researchers like to have that sort of information. But as soon as samples enter the biobank, all identifying information is removed from the sample. So that includes things like name or date of birth. And this is to protect the privacy of the donor. And all the clinical data is stored in a HIPAA-compliant manner. The goal of the biobank is to advance EHE research and allow investigators the ability to request these rare specimens for their experiments. And in this way, studying the disease, creating research models, and testing drug efficacy is done directly with human EHE samples. And we believe this is crucial because in many cases, like Dr. Rubin and others have mentioned, animal models like mice or rats are not very representative of disease states. However, the mouse model that Dr. Rubin published on in 2021 does look like an excellent preclinical model, as do the PDX models. But usually we think it's best to just have sort of a lot of tools in your tool shed to investigate, disease mechanisms and possible treatments. So why do we need the EHE Biobank? In many cases, even researchers associated with large hospitals do not have access to fresh EHE tumor cells, simply because there's not enough EHE patients coming through their clinics that are associated with the research labs. Our biobank provides access to those sorts of specimens. And having a centralized repository makes it easier for those labs to get access to those specimens. Because these samples are from a wide range of EHE patients, it allows scientists to study the heterogeneity of the disease. There are many ways that a researcher might use the biobank samples, but a great use would be to create research models, like the cell lines that Dr. Tanas created. Another one would be the PDX models that we just heard about moments ago. And I'm not going to go into how those are generated because we just heard about it. Another great use of the biobank would be to use the samples in it to research biomarkers that could monitor disease progression and severity in patients. By donating samples to the bank, patients can help drive advances in EHE research, and the more samples that we can offer scientists, the better their investigations will be, and the more advances we'll have in patient care. Not only that, we like to think that it fosters collaboration between research groups, which is an excellent driver of innovation. So how does the biobank work? It begins with a patient or physician contacting the foundation directly and indicating the willingness to donate a specimen. Patty and or I will obtain the patient's signed consent to participate, collect some basic clinical information, answer any questions about the process that you may have, and then we take it from there. We contact your care team and organize the logistics of collecting the sample. The sample is then sent directly to the EHE Biobank, which is housed at the Cleveland Clinic Central Biorepository, where the tissue is processed, analyzed for quality by Dr. Rubin, and stored. Researchers can then request access to the samples by contacting the EHE Foundation. And because these samples are so rare, the research projects are reviewed before specimens are distributed, just to make sure that the samples are being used in a meaningful way. But once it's approved, the Cleveland Clinic will send the researchers the requested samples. And you might be asking what we've been up to since 2022, and a lot. So Patty, and to a much lesser extent, I have consented 82 patients. We've collected tissues from 47 different cases. We have 25 active cases where we're collecting what's called retrospective tissue, or tissue that is stored in a hospital from a previous surgery. We've distributed 21 specimens to 4 different labs. And most excitingly, we have 4 research models that are in development and nearing readiness for distribution. I'm going to go through this kind of quickly because I'm running out of time, but this, I just want to show you the diversity of what we have in the biobank currently. So we have a wide range of coverage of different types of patients who have donated to the biobank. If you're a researcher and you're interested in obtaining EHE samples, we have them processed in a number of ways and can likely meet your research needs. We currently store blood samples as plasma and packed red blood cells. We have fluid from ascites and pleural drains. We have cryopreserved tissue. We have snap frozen tissue. We have formalin fixed tissue, and we have pre-cut slides. We try to prioritize the collection of fresh tissue, which is processed on site at the Cleveland Clinic by Dr. Rubin's team. And currently we have over 650 individual samples in the bank, and we're always excited to add more. We're actually enrolling participants with an emphasis on creating cryopreserved samples with matched blood samples. So if you're a patient and you're interested in donating tissue, please let us know. If you have an upcoming procedure, the earlier we're notified, the better. It can sometimes be a little tricky to coordinate with clinical teams about the donation. We can also arrange for fluid connections and post-mortem donations. Importantly, please keep in mind that this will not cost you, the patient, a penny, and we make sure that your identity and privacy are protected. Any donation that you might make is going to directly benefit the entire EHE community. I'd also like to highlight that if you do donate, and then down the line decide that you no longer like to be part of this initiative, you can just let us know, and we will destroy all physical samples within our biobank, as well as any clinical data that we might have, and we'll instruct any research labs that we've distributed the tissue or information to do the same. And if you'd like to find out more, just give us an email at biobank@fightehe.org or visit our website at fightehe.org/EHE-biobank. And very quick acknowledgements, because I want to get to the Q&A. Thank you so much to all the past and future biobank participants. This would not be possible without you. And a special thanks to those who keep the bank running smoothly, namely Dr. Rubin and his team at the Cleveland Clinic, who are instrumental in collecting and distributing the samples. Jane, who did a lot of the legwork on starting up the biobank and still helps us connect with donors. Patty, without whom none of this is possible at all. Dr. Tanas, who provides invaluable guidance and has developed the cell lines that you saw earlier today. And the North Star Independent Review Board, who reviews our protocols and amendments and makes sure that we're working ethically and securely. I'm happy to take any questions in the Q&A, or I can field questions offline if you'd like to email me at sethhaddix@fighthehe.org. Thanks so much for the opportunity to speak to you guys.
If I can welcome back Dr. Tanas and Dr. Pasquali, we'll do a Q&A session with the first session group here. And we're also being joined by John Lamar, who will participate as a Q&A panelist. And we have a question from the audience, and this might be best directed to Dr. Pasquali, but welcome anyone to answer it. Question is, based on the results you reported, do you have any feeling for whether sirolimus may be more effective in aggressive EHE cases relative to non-aggressive?
Yeah, thank you for the question. I think this is very interesting. The definition of aggressive cases is about cases that actually progress or have symptoms. So I think that our medical oncologists mainly use sirolimus in those cases actually, where there's definitely the need of a treatment or at least stabilizing the disease. But probably tomorrow medical oncologists could answer better to this question than me.
Any other thoughts on that question from anyone on the group here? And so as I understand the answer, this is normally going to be administered to people who would be sort of on the more aggressive side. Yeah. Based on your results, is there any suggestion that sirolimus or a drug like sirolimus might be used to in patients who are not aggressive yet as a way to prevent transition to an aggressive state?
From my perspective of a lab scientist, this is very difficult to assess because our model is based on a very aggressive type of EHE. Probably like the models that Munir showed before, that are primary cultures and derived also from patients with less aggressive disease, might be to some extent very interesting to assess this because the molecular profile of tumors, of less aggressive tumors treated with sirolimus could be maybe investigated. So I think this is a very interesting question and very relevant, but it's probably need to be tested in other models as well.
And then we have one more follow-on question on the same kind of topic here. We got a question from the audience asking whether everolimus, or maybe we could apply this to any of the other similar sirolimus-like drugs. Is there any feeling for whether one might be as good as the other?
I don't think we have a comparison on this, so they should be very similar.
And I know some of them were developed with the potential to have sort of less side effects than others. From the molecular mechanism, you think they would be expected to be fairly similar?
Yeah, absolutely, yes.
Okay. All right, let's a question for Dr. Haddix. You described in, I think, very efficient detail kind of what the EHE Biobank does and how people can contribute samples. And I couldn't help but connect the dots from what everyone's talking about here. It sounds like with Dr. Tanas, generating these cell lines. Can you describe a little bit, is there a direct link between the biobank and the work that like Dr. Tanas is doing?
Yeah, it's a direct link. So Dr. Tanas requested, tumor samples to derive his cell lines from, and I didn't provide that. Patty Cogswell and Denise worked on that. I've only been working for the foundation for about 4 or 5 months, but there is a direct link. And so the idea here is to create collaborations between the foundation, the biobank, and researchers, just like we have with Dr. Tanas, to create more models and spur more experimentation and looking into disease mechanism. And another thing that we think is very important is looking for easily accessible, non-invasive ways to assess disease progression, like Dr. Rubin mentioned. So blood draws are quite important for us.
And then maybe a follow-on to that with Dr. Tanas. I mean, you in your presentation, you identified 4 lines that you've been developing. And I know this is still like an early stage and this is all sort of new, exciting work. based on what you've seen with those four, do you have a feeling for, what is your sort of level of optimism that this is a pathway that's going to work if samples become available from patients to be able to get more than those four and to sort of reproduce the work you've already done?
So we've since then obtained a fifth sample that we've been able to grow out the same way, and we're characterizing it as well. I think it's a reproducible approach. I didn't show, after I finished the talk, there was one other piece of data I want. Can I share a screen real quickly? Is that possible?
Yeah, I think you should be able to share here, and we'll all still be live.
Yeah. So this is single cell data from EHE-01. No, this is looking at, one of the questions that comes up when you make these extended primary cell cultures is regarding the heterogeneity of the culture, and this is single cell data looking at CAMTA1 expression and FLY1 as surrogates for expression of the fusion and endothelial marker, and you can see here that the cell cultures are very homogeneous, and and that in conjunction with the fact that we were able to do this with a fifth sample, I think this will be something reproducible. We've actually put these findings up in bioRxiv. We're submitting it for publication now, so other folks can grow cultures out the same way for their own use. And we're also working with Seth and Patty and Denise to make these available to other investigators. We're working with a third party to bank these cell cultures. So I think that this will hopefully, all these. It's another model system. I think in conjunction with other model systems that have been developed, I think these will be helpful.
Excellent. And we've got a couple more questions from the audience. I think these are best for Dr. Haddix. The first question is, how many times can the same person donate tissue?
As many times as they want. So anytime you're going into surgery, if you contact us, we're going to do our best to collect any excess surgical tissue that the hospital might just throw out if you don't contact us. So anytime you have a surgery, give us a call, and we'll do our best to get that your donation into the bio.
Excellent. And another question for you. Is there a need for blood samples if no tissue is being collected?
Yes. We will take essentially anything, but blood, even without a matched tissue sample, is very valuable. Absolutely.
And then we've got a question for Dr. Pasquali. Would you consider the increase of GDF-15 level as prognostic or diagnostic for EHE? And then I'm going to ask a follow-on to that. If the answer is yes to that, what might this look like in the near future about how this could be used with patient care?
Oh, thank you. This is a very interesting question. I think that GDF-15 could be used as prognostic. Not sure about diagnostic, because there are tumors that show there are other tumors that can show high level of GDF-15, such as osteosarcoma. So it's not entirely EHE specific. Maybe I misunderstood something before. Compared to other tumors, GDF-15 is very characteristic of EHE, but other tumor express. In terms of further developing it, we are working on that to try to follow a path of a biomarker approval. But it's a quite there are a lot of hurdles, and at the moment we are working on that together with the foundation and Royal Marsden and ICR.
Excellent. And we've got a question for Dr. Lamar, and I'll also then use this as an opportunity to see if Dr. Lamar has any questions for the group here. The question for Dr. Lamar, and this is a broad one, what do researchers need that they don't have?
Yeah, I mean, that's a great question. I think starting with a biobank is a good place. What cancer biology has taught us in the past is that you really need lots of examples of patient tumors. You need lots of blood samples to really make sure that the things that we're discovering in the lab can translate into the greatest number of patients. And, we only have a couple of human PDX lines so far. Dr. Munir and Pasquali both talked about these, but the more we have, the better we can understand the discoveries that we're making and how they're going to relate to all patients. And so I think donating tissue, donating blood is a huge first step. The second big step is that we need research funding. And I think at a time now when sort of research, especially in the United States, is potentially being impacted by political changes, I think foundations like the EHE Foundation are going to be really the main funders of EHE research moving forward. And so any donations that can be given to the foundation will allow researchers to keep their research going as things get tougher, sort of from the political side of things. So I think both of those things are really important and things that patients can do and patient advocates can do and family members of patients can do.
Excellent. And then did you have from what you heard from the panel here, have any questions you have for the group here?
I don't, nothing specific. I think there was a question in the chat really about sort of things that can be used to predict progression. And so I guess both for Sandro and Munir, do the human cell lines have tumor suppressor loss in the ones that you've looked at, CDKN2A, RB, or P53? Have you seen those in the more aggressively derived lines? I'm just curious.
I start with, for instance, our cell line have a loss of CDKN2A, so that's for sure, and also RB.
Yeah, I think we saw a SNP in CDKN2B, and then CDKN2 and P53 the expression does go up with continued passage. I mean, it's clearly an obstacle to immortalization or in these lines, and so I suspect it would be associated with more aggressive ones, but we don't have definitive data at this point.
Great.
We have a couple more questions that came in related to patient donations. First one, I think, is a quick one for Dr. Haddix. For the biobank, can any patient at any time donate blood without having another procedure?
Long and short of the answer is yes. So we're currently trying to get a program initiated where a mobile phlebotomist could come to your home and draw your blood and get it to us. We're not fully online yet. We're doing test cases, but hopefully soon we'll be able to do that. We've done it for other, Patty and I have done it for other biobanks, and it's been very successful, and we've got a lot of community buy-in.
Excellent. One more donation question here, and this is probably for Dr. Haddix and Dr. Pasquali, because there's an international and US aspect to it. So it's the first part of it is, can a post-mortem donation be arranged? But then the question says, especially if you're living outside the US, which maybe reaches the more general question about geographic boundaries and how tissues are donated in different areas of the world.
Speaking from my experience, it's relatively difficult to get any donations outside of North America to biobanks situated in North America, especially like the EU or China. It's not to say that we're not going to try. So there's always a way to do something, and we're gonna do our best. But knowing that Dr. Pasquali has kind of a pipeline, it sounds like, for donations, maybe you should speak on it more.
Yeah, it'd be good to hear what's sort of the European options available.
I've been working a bit all around the world, and I think there's a different perception of this in different countries from patients and their families, as well different legislation. So it's very important to address this at the local level. In some countries, in some culture, is definitely an option.
Very good. And there's a few more little questions that were in the Q&A, so I encourage the panel here to, if you have the time and willingness to do so, take a look at those, and maybe we can answer some of those in writing. This is the end of session 1. Thank you all for participating, and we're going to turn towards session 2.
Thank you.
It's been a pleasure.
Thanks.